Here in part 3, let's talk about some structural changes that were required for plants to get established on land. One of the key features that we see is the evolution of structures such as a cuticle and air pores. So a cuticle is a layer of wax that's on top of the epidermal layers, and it has the function of retarding water loss. It also retards carbon dioxide uptake, but its primary function is to retard water loss. How did CO2 get inside to the photosynthetic cells if there was a cuticle? It was through pores, openings, air pores that were uncontrolled, simply openings. Those are called air pores, and you see a cross-section here of an air pore consisting of about 8 to 10 cells in a liverwort. That allows carbon dioxide to come in and allows water to leave. So in an environment like that, if there's no regulation on water leaving, then these plants require a moist surface or surface that had adequate water all the time. When we begin to look at these plants, they lack leaves, they lack stems, they lack roots. They have no vascular system, and they have no ability to control water loss. So carbon dioxide moves in through diffusion, but so does water. So these plants are only able to persist in environments where there's adequate surface water. At times, when there isn't adequate water, these plants desiccate, as I had mentioned in a previous slide. So this is the capacity to go from being active in a wet, hydrated condition to being totally dry. Here you see a liverwort in a hydrated form and now in its desiccated form. It's still living, but it's desiccated, it's stabilized in a dry condition. That is a feature of early land plants that is no longer retained in the more derived plants. After you add water to them, they quickly rehydrate, protein synthesis takes place again, chlorophyll function resumes, and they can photosynthesize and grow. So let's have a clicker question. What adaptation is not found in liverworts? Sporophyte, vascular tissue, cuticle, or air pores? So if we begin now to look further into the key adaptations in the evolution of land plants, we see that the first feature that separated the algae in aquatic environments from the terrestrial environments was the presence of a cuticle on the surface and air pores. The second feature which then evolved was the presence of a stomate. And a stomate is a pair of cells. They're called guard cells. They come together to make, the guard cells are kidney-shaped. They come together to make an opening. And by changing the size and shape of those guard cells, the stomates effectively open and close. So the first adaptation to land is a cuticle and air pores. The second evolution is replacing the air pores with stomata. This happened in the sporophytic form of the moss and all subsequent plants. We tend to use two terms, and it doesn't matter which one you use. We can say stomate or stoma, the plural being stomates or stomata. They open when in the presence of high light. They close in the absence of light. They open in periods of adequate water supply, and they close to save water when water is in short supply. So this was a key feature allowing plants to regulate their degree of hydration. As I'd mentioned before, leaves have a cuticle, and that cuticle prevents water loss, but it also prevents carbon dioxide from coming in. So the evolutionary trade-off that plants have is to take up CO2 while minimizing water loss. Our own Bill Andrick at the University of Utah is one of the theoretical leaders in describing how plants regulate carbon dioxide uptake versus water loss from an evolutionary and ecological perspective. In order to change the diameter from being closed to being open, basically potassium ions are pumped in and water follows through osmotically to expand the size of these kidney-shaped cells. It turns out if you look at the cell wall thickness, here on the interior portions, the cell wall is much thicker, so that you don't have an equal expansion. What you have is these regions resisting expansion and the expansion taking place there. So we go from a kidney-shaped closed to a kidney-shaped open, all by taking up the potassium and chloride ions. Stomates first appear in the multicellular sporophyte attached to a gametophyte. So here's a moss in its typical condition. That's the gametophytic or haploid condition. There are no stomata, so there's no regulation of water loss. But the stalk-like structure here is the sporophyte. This is the 2N form of the alternation of generations. And it's in this form that we first see the presence of the stomate. Why? We don't know, but we can speculate. And one speculation is that the rate of water movement through capillary action from the gametophyte to the sporophyte is slow. And to maintain hydration, then the sporophyte has stomatal pores that close that reduce the rate of water loss, allowing hydration of the sporophyte. Clicker question. Point to the diploid portion of this plant, and you should be pointing here. The sporophyte is the diploid portion of the alternation of generations, and the lower part is the gametophytic or haploid form. So in a moss, as in algae, most of the life cycle is spent not in the sporophytic phase or a diploid phase, but in the haploid phase. Mosses have that same alternation of generations as we had shown, going from male and female gametophytes, where water is used to transport the sperm, to the production of a sporophyte. That sporophyte disperses the diploid form, which then undergoes meiosis to create haploid spores. Those haploid spores can now germinate. The advantage of the spore form as an intermediate, whether it's a diploid spore or a haploid spore, is that it's able to persist through a drought period until water comes back. Clicker question. Which adaptation is not found in mosses? The sporophyte, the cuticle, the air pores, or the vascular tissue? I think I've given away the answer here. It's, of course, the vascular tissue. Vascular tissue is a more derived feature not found in mosses, but mosses do have a sporophytic portion, they do have a cuticle, and they do have air pores. So what are the advantages of height? What are the problems associated with height? What are the evolutionary solutions? And going from the very prostate liverwort form to the more elongated and taller moss form, there was a change in height, It had some advantages. One is greater dispersal of the gametophyte. Second is the ability to compete better for light. But there are two disadvantages to height. One is that you need to move nutrients from where they're acquired here at the soil up to the photosynthetic tissues. And you need to be able to support that structure without it drooping over. So that required some evolutionary changes in order to go from being prostrate to being tall.